I really respect all the people I've met so far, and we've already had some amazing conversations. And so to give you just a little background, I'm a traditional developmental biologist and animal behaviorist. I grew up collecting caterpillars. I studied them in grad school for my PhD. Developmental biologists are incredibly interested in this question of how you build form and function. So really, the question is, how do you get from an embryo or an egg to a really complicated creature like this? And obviously, insects are super cool for this question because there's a second form of transformation that happens, which is metamorphosis. So this is a crawling animal. It chews plant material. This is Manduca sexta, so if anyone here is a gardener and you get these big green caterpillars that eat your tomato plants, this is the organism that's studied. But this goes through this incredible transformation to this night-flying hawk moth. And what you see here is this incredibly long proboscis or tongue. So there's a number of flowers that only this species of moth can pollinate because it has this really long tongue. It can go in and get the nectar. It flies, it has complex eyes, and it has incredibly sensitive antennae. You basically have to rebuild the form and function. So as a developmental biologist, part of what I did is try to understand the molecular cues that happen to get these cells to coordinate during these events. And I also looked at the brain to see if memories could persist between these two stages. So can you teach a caterpillar something that a butterfly remembers after undergoing this incredible transformation? And as biologists, we generally look at this from the point of view of the tree of life. We're interested in understanding how you generate form and function in the extant species that we see around us. And so really, you're in this purview here of looking at existing, typically multicellular, sometimes unicellular lineages, and how do you connect all of these pieces in this fitness landscape to generate a functional animal? The hard question is, how do you do the inverse? If we have all of these pathways on how to generate form and function, can we design it? Can we create shapes, anatomies from scratch that we may be interested in as humans to do work or to enhance certain features, or also in the realm of regenerative medicine to regrow lost parts? We've done this the hard way, which Sam has already brought up, where we can take existing things. I believe this is called teosynty. Apologies for any agricultural historians in the audience. This is the common ancestor of corn. And through several thousand years of selective breeding, we've created basically a biological machine that feeds us. And on the left side, of course, we've domesticated huge numbers of dog species from the wolf, but we're still really limited by this space of what exists. We can't design outside of the realm of things that evolution has already provided previously. And that's really interesting because we would love to be able to build from the ground up. And in biology, this has proven incredibly challenging. So really only in the past years has biological design made progress. And this generally falls into three categories on the biology side. So synthetic biology, biomaterial science, and bioengineering are all cracking away at this problem. And really at the molecular level, can we design form and function in a way that is now outside of the traditional evolutionary history? So the things that they're building do not fall on this evolutionary graph, and as Sam said, you can transverse this fitness space very easily. But for me, as a biologist, what's been hard to learn is I never talked to roboticists, but this has been the wheelhouse of their discipline for the past 75 years. They're incredibly good at building form and function. They've learned models, they've built simulations, and they've built physical instantiations of these. But these two groups have never met. There's no reason, except for conferences like this, that you might overlap. And so those conversations just happened spontaneously through a grant that Sam and I were part of, where we met each other and started talking. And the idea was, could we combine these principles? And so this isn't our work. Originally, there's a lot of people taking this sort of approach. But really, the goal is to take these two things and dovetail them together to form what's been colloquially called to as biological robotics, or perhaps more broadly, to build autonomous living machines. I wanted to also start by backing up. You've seen a little bit of this, but as a biologist, when I think of a robot, I think of some hard, rigid thing that sort of walks around, and you know, the best state of the art is still fairly frail when it comes to the types of things it can do, but robots are formed out of all sorts of different things. So this is a soft robot that I really like. This is similar to what Sam showed you. It has actuation in the front, middle, and back, where it just pumps air into a soft body. And it's able to do all sorts of different behaviors that a traditional hard robot can't do. So you can tie these in a knot, you can send them down drains. And this is absolutely a robot, and there's a whole field of soft robotics that looks at these soft materials. And so I just am leading you through this because it took a lot of work for me to think about what is a robot, what can it be made out of? And it's not really material dependent, it's on how it's programmed and what it does. This is one of my favorites, so I won't make any claims that this is a good robot, but this is a robot that's made out of pine cones. And so these are just pine cone pieces that you can collect yourself and have been glued together with a hot glue gun. And these serve as hygromorphic actuators. So when you get this material wet, it swells and it allows this to move up. And when you dry it out, it expands and it goes back down. And if you build these different designs, you can create really simple types of walkers and machines that look like this. And this is obviously sped up a lot. But again, this is a robot built from a biomaterial. So this is something produced by plants. And there's a bunch of different versions of this. They're even trying to make walls that as they rain, they'll open up and close to sort of autonomously provide some sort of function without any electricity. So where does the biology really come in? Using biology in robotic design, this typically falls under what we would call biohybrid systems. So these are combinations of living cells, which are typically used as motors, and synthetic materials that are typically used as scaffolds. And these are just 3 of my favorites out of a huge palette. What you're seeing on the left is a view from the bottom, but this light material is just basically a plastic or rubber backbone. And these dark bands that you see running around are rat cardiomyocytes that have been grown and attached. And this is in water, and if you pulse electricity, it causes these to contract just like our heart does, and this can create a very simple linear motion. And this is one of the first ones. This is from the Asada lab. Just down the street in Boston is Kevin Kit Parker's lab. He is an amazing scientist, and he's been building some really cool bio-inspired designs. So this is a bio-inspired jellyfish. This is synthetic backbone, again, a really thin piece of cellophane that's carefully seeded with muscle cells in particular orientations. There's some beautiful molecular biology that goes along with this. Again, by pacing current, you can get this really incredible swimming design. And he works on the physics of these systems and the stresses. And last, we have things that are more typical biomachines. So here in white, you see opposing, these are skeletal muscle bands, and they're between electrodes. And as you pass current back and forth, you can get this arm to articulate and perform very simple types of work. So why would you want to build this? There's a bunch of reasons. Number one, you can actually learn a lot about the biology of what's happening by learning how cells self-assemble and self-organize. And on the other hand, the biology is great because, number one, there's a huge amount of biochemistry, like Sam said, inside of every cell. We're not really leveraging it in the designs that you see here. But also, the biology is biodegradable. And so if you were to build a fully biological version of these, you could put them in environments where you'd be worried if a traditional robot ceases function. So a very sensitive aquatic ecosystem inside the human bodies, inside microfluidic devices, and so forth. When these cells degrade, they break down harmlessly. Animals die in the water all the time, and basically the microbes clear them out. And there's much less risk of things that have batteries, wires, and plastic materials. So this is where my research really combined with Sam. We were looking at some of these and thinking, could we replace these synthetic parts and just make a fully biological version? And so how do you do this? And this is really on my background. Developmental biology has never been really integrated or dovetailed with traditional robots, but we have all the tools. We have 100 years now of stem cell biology that gives us this rich tapestry of palettes with which to build. And so I use Xenopus laevis. These are amazing frogs. They live in the lab for about 20 years. A female frog will lay about 3,000 eggs every 3 months. They're external, so you have this incredible source of stem cell material from which to collect. And if you take some eggs and you have them in just tap water and you leave them on the bench, after about 24 hours of development, you have about 4,000 to 6,000 cells. And these cells are all stem cells, so they can become any cell that you would like. But we have what's called a fate map. So you can project on top of this like a globe. And we know that certain regions natively will become different types of tissue. So if you can't read what these cells are, these green regions will become skin. These purple regions will become neurons, and this yellow region will become the internal organs. Again, we can push these and deflect these, but natively, you have all of these rich pieces of tissue. So you could use a chemical treatment and basically dissolve this organism as it's forming, and this gives you a bunch of Legos with which to build. So you separate out all the green cells, the purple cells, and the yellow cells, and then you can recombine them and reconfigure them to build a geometry from the ground up that does not exist in nature. So this would be something that has skin cells on the top and bottom, and the red is a band of muscle in the middle. And as a biologist, this sounds quite strange to deconstruct an organism and reconstruct something new, but this is really where the roboticists provided insight. There are lots of robotic systems that you can disassemble into scaffolds, motors, sensors, actuators, and control systems, and build something completely new from those parts. So again, a robotics approach to biology. This was a huge frame shift for me. What does this look like, and how do we know what to build? Sam already provided the background, so I'm not going to go over that. But we have this huge space of things to build, and it takes me a long time to build these. And so Sam uses a simulator to simulate these different cell types, and he can run through thousands or millions of designs in a month, and he can give me the runtime champs. I can maybe build 30 in a week. So if you talk about accelerating research and reducing bio-waste, the simulator is great. And those are then supplied to me from the computer, and I build them with my team in the wet lab. And I know people, for some reason, love these how it's made videos, so I just wanted to give you the super short version. We're cutting out a whole lot of the work. But really, this begins with these frog embryos. So these are about 1 millimeter in size, and this is me using a very small pooled glass capillary to deliver RNA or DNA to these embryos. This can change gene expression or allow me to do lineage tracers. After 24 hours, I collect these different bits of the embryo, and I have to clean off this outer bit of epidermis. It basically gives me these fluffy white stem cells, and I make different piles and collect them. And then from those piles, I make wells that are about 0.5 millimeter large, and I rebuild the anatomy from the ground up. So I layer these cells almost like building a sandwich and allow them to come up. I swap out the media, which allows everything to heal together, so this locks in the anatomy that I've built. And that takes about 12 hours. And then on Friday of every week, I sit at the microscope with a really fine cautery electrode and some hand-polished forceps, and I sculpt away the negative material to match the design that Sam has given me. So this is something that, as he showed you, was about half of a poppy seed, so this takes a lot of fine motor control. We're trying to automate this process to increase the speed at which we can go, but what comes out the back end is a completely stable, micro-sized biobot. This is really amazing because for two reasons. Number one, since these are sourced from amphibian embryos, they live in tap water and can survive from 4 degrees up to 24 degrees Celsius. Number two, because they're embryonic, they're loaded with yolk from the maternal embryo source. So these are self-powered for 14 days. You can build them, their geometry holds, they behave, and you have to do nothing. You could just essentially put them into up to brackish water. And so this was really just building the shape. On the left, this is what Sam showed me, and I told him I could probably build this with cells. And I believe there was audible laughter from him, and so as a biologist, that's the challenge. And in 10 days, this is what I came back with. We have a Slack thread where I first showed him this was possible, and this was really the genesis of the project. And as he showed you, we don't need to talk about it, but it's not just the shape. I can match the cell identities. This is staining for these 2 different tissue types. And so this is matching the form from the point of view of biology, and then also we've matched the function of what's been designed. So he showed you this video before. This is a side or lateral view of these contractions in real time. So this is a completely synthetic built system. And this is a top-down aerial view that's been sped up 4 times of this linear locomotion. This type of muscle cell is a really great actuator, but it's limited in the fact that it's binary. Muscles contract or not. You can't really half contract a muscle. You can half contract maybe bands, but you're limited in control. So one of the other things we've done is expanded our actuators to cilia. Cilia, like on the inside of our lungs, are a type of propulsion that dominates the microscopic world. So if you look at things in pond water, they often have these small hairs on their outside that beat. And molecularly, we can add them or remove them for the surface of the biobots that we build. And this generates different types of motion. So this is a very high magnification image of these tufts of hair that are beating. This has been slowed down about 10,000 times. This happens really fast. But this is designed with no muscle on the right. That's this donut, and it's propelling itself through the water just based on the coordinated beating of these cilia patches. And if you tweak the anatomy, you tweak the cell types, and you tweak the actuators, this gives you this incredibly rich design space with which to design these biobots. See, these are just some that have been built in the lab. This has a combination of muscle cells and cilia and is moving. You can compress these down, so this is one that's only 2 cell layers thick, so we have some applications where you might want something that can move under very small obstacles. This is the donut in high magnification that you've seen before, and again, this is just another type of thing that I've built. And so where the research really has been at is sort of at this phase. We're sort of expanding the palette of usable tools. We're adding more cell types. We're trying to get some sort of more interesting behaviors and learn some more about the biology. And we have sliding design from basically a completely non-shaped just ball of tissue like an organoid to a really complicated and highly structured 3D biobot. I just want to leave you with maybe 2 notes on next steps and sort of the bigger picture. One is, obviously, one of the things we're doing right now is we have very little control over the system in real time, so I'm actively investigating controlling behavior of individuals in swarms, and we're looking at the different types of environments we might be able to exploit. And so these are aquatic mazes that this one's about 3 millimeters wide, and this is a capillary tube that's about 0.5 millimeter in diameter. So these are environments that would challenge traditional robots at this size, especially an aqueous solution, but we can build things that can easily traverse these. And on the swarm side, we're looking at behaviors like particle aggregation, particle dispersal, carrying particles around, delivering cargoes, and so forth. And on the other side, I just want to loop this back to what I said at the beginning. I'm not a bioroboticist, I'm a developmental biologist. And by putting these tissues together in novel configurations, we've learned a huge amount about the self-assembly and self-organization of disease-relevant tissues. So a lot of the tissues that I use are models for human airway disease, and we've learned some really fundamental things that we wouldn't using these traditional developmental biology approaches. So for me, the excitement has been we're sort of getting the best of both worlds. We have an applied wing of the research program and a basic science research program as well. And I just wanted to finish on maybe an inspirational note that's not directly related to the science, but as someone who's been in the field for about 15 years, this was the first research I ever had that sort of went viral, so to speak. It blew up. And it went out into the world, and these terminologies that we find so sacred in biology mean different things to the public, which was the most anxiety-inducing experience of my life. And so suddenly you've lost control of the argument of your own research, right? So this is a YouTube video, this is a living robot, not my words. Scientific American did a special on this that's called Animal Robot. And I think engaging with the pop press is something that I'd never learned in science communication. And for any young people who are maybe this happens to, I want to say yes to these opportunities, and that anxiety is not worth it for what you get out of it. So I was afraid of them getting the story wrong. I was afraid at my research losing control of the story. But I want to say, on the other hand, a couple things. Number one, the public and all disciplines are stakeholders in how our research impacts the world. They deserve to have an opinion and have their opinion be heard. Number two, this helped me learn scientific communication in a way that I never would have been able to. Interacting with these people was hugely important. And afterwards, my parents were suddenly like, I actually understand what you do now. And that was an important lesson to learn and incredibly humbling. And last, what I resist is obviously you don't want to hype your research, and we resist that as scientists. But what you do need is you need to have someone see your research. You can paint the most beautiful picture in the world, but if you hang it in your closet, I don't know that the utility is there. And these experiences of interacting with the popular press have brought new collaborators to me that I would have never met otherwise, just because they're in disciplines that would never paid attention to this research. And I just wanted to finish that saying, while the museum exhibits are just super cool for me as a person, these institutions are actually critical for interfaces between art, science, and society. They understand how to engage the public, how to market things, what's going to make people afraid, how do you touch things, how do you get young people excited. And so if you have local museums in your area, don't feel scared to reach out to them and say, hey, I do this really cool research. Could we build an exhibit, or could I come and talk to your people? And they have been instrumental in helping me as a science communicator, really working with the general public and learning how to frame this in a way that makes it palatable to society at large. And so I think I'm right on time. I want to thank all the members of my center at the Allen Discovery Center, Sam and Josh Bongard and Rebecca for the great introduction, the funding sources, and especially all the people here. I am super excited to have gone first because selfishly it gives me the most amount of time to interact with you. I'd love to talk and hear about what you do as well, and hopefully this can be the beginning of a better conversation. Thank you.